Let's talk about dominance. Now, dominance is the phenomena that was observed during Mendelian experiment when Mendel was studying the hybridization experiment involving contrasting traits. So, as per the Mendelian concept, the character that is going to be expressed in F1 generation is actually known as a dominant character, and the character that remains suppressed is known as a recessive character. I am sure you know that if I cross tall with a dwarf plant, in F1 we are going to get tall plant, right? So what we are seeing, that this tallness is going to dominate in the F1 generation and this dwarfness is going to remain suppressed. So whatever the character that is expressed in F1 generation is actually called as a dominant character. So please remember that dominance is always going to be identified with reference to F1 generation. Now there can be different type of dominance like codominance, intermediate dominance, pseudodominance. So we will talk about the different type of dominance, right? So have a look at the very first, what we call intermediate or incomplete dominance. So here what happens, that a new phenotype is going to emerge in F1 other than the parental one, because in the previous one what we have seen, if tall is going to cross with a dwarf, either tall or dwarf is going to appear. But whenever there is going to have a new phenotype emerges which is other than the parental one, we call them the intermediate dominance or incomplete dominance. Okay, and here F2 genotypic ratio is typically modified into 1:1, because we know very well that phenotypic ratio, a typical phenotypic ratio 3:1, instead of 3:1 we are going to get 1:2:1, right? The perfect example is Mirabilis jalapa, and in case of Mirabilis jalapa there are two types of flowers. The plant produces two type of flowers. One is red and second is white. Okay, and suppose R is the gene that causes red, so capital R can be written for red, right? And similarly small r can be written for the white one. Okay, so they are going to produce only one type of gamete and they are going to produce also one, that is small r, and the F1 we are going to have R and R. So instead of either red or white, it is going to have of pink color. Okay, so this was your F1 generation. And if I go for a Punnett square, in case of Punnett square what we see, that there is a deviation from Mendelian ratio. Have a look at this. So I am keeping the gametes in the Punnett square. So we are going to get capital R, small r, capital R again is small r, and small r. So this is going to be red, but this is going to be pink, right? Here it is also going to be pink and this is going to be white. So what we see, that genotypic ratio is same as to the phenotypic ratio, and that is equal to 1:2:1. So this is about the intermediate dominance, right? Now have a look at another type of dominance, what we call codominance. Now codominance are such a dominance in both alleles are going to express equally, right? And if it is going to happen in F1 generation, we call them codominance. So in this case also what happens, that genotypic and phenotypic ratio is modified and both are 1:1, that is as observed in case of incomplete dominance. The typical example is Rowan cattle. Now what is Rowan cattle? You see the cattle which is having a white color as well as the red color fur or coat color, we say is a Rowan cattle. Okay, so let's see that how this Rowan cattle is actually being formed, right? So suppose a red color cow, right, that is going to cross with a white color ox. Okay, so again it is going to produce only one type of gametes, and it is also going to produce only one type of gamete, that is small r. And if I cross, then what we are going to get, capital R is small r, right? And this is your F1 generation. Okay, and this is what we call is a Rowan cattle. Why we are calling Rowan? Because here what happens, that this capital R and small r both are expressing itself. It is not like that R is dominant over a small r. So R was responsible for red fur and small r responsible for white fur, right? So Rowan, they have a both white plus red. So here both white plus red are actually present. So that's why we are calling them Rowan cattle. Okay. So again if I go for, let it be, so Punnett square. So in case of Punnett square, again we will see that there is a deviation from a typical Mendelian phenotypic ratio of 3:1, So have a look at this. So just now I am keeping the gametes like this. Okay. So in the first we are going capital R, this is going to be red, right? Then we are going to have small r as well as the capital R, this is going to be ρ1 as we have discussed, and again we are going to have r1, but small r that is going to give the white one. So again we see that phenotypic ratio is same as to the genotypic ratio, and that is equal to 1:2:1. So here the typical law of Mendel is actually not being followed, that is law of dominance is actually not being followed. So that is why. So once again I would like to reiterate that in case of codominance, the alleles are going to express equally. That is the main thing as far as the codominance. Now have a look at one more type of dominance, what we call pseudodominance. Now in case of pseudodominance, what happens when recessive allele in the absence of its dominant counterpart start behaving as a dominant allele, we'll call them pseudoallele or pseudodominance. And it often occurs due to mutations. Okay. So have a look at this. Suppose there is a homologous chromosome like this. Okay. And there are two locus. Let it be that capital R, small r, and T and small t. So the phenotypic expression of this particular plant is what we can say tall red, right? But suppose due to mutations, what happens? That this capital R gene is lost due to mutation. So what will happen? In that case, it will be like this. So small r, small t, but here is a capital T. Now the phenotypic expression of this particular will become tall white. So here white is going to behave as a dominant allele due to absence as counterpart. So this is all about the pseudodominance. The perfect example is seen, that is hemophilia in males in case of the human, because we know very well that humans have a XY chromosome. X don't have any counterpart, right? So that's why if anybody is going to behave, let it be say a gene for hemophilia is actually present over there, then it is going to express. That is in case of male. Why? Because the dominant gene is not there to suppress it. Had it been in the case of female, what will happen? That this X which suppress it, the laws of dominance or the phenomena of dominance will be there, then female will not be hemophilic. But in case of male, so he is going to be hemophilic. Why? Because this is showing the phenomena of pseudodominance. Okay. So let's conclude the entire concept of dominance. So we can compare dominance, codominance and incomplete dominance, right? So if I talk about the dominance, in case of dominance, if I take a heterozygous condition, let it be like capital T and small t, here what we see that it is going to express 100% and a small t is going to express 0%, right? In case of codominance, what will happen? If I take the same example, then here it is going to express 50% and it is also going to express 50%. But if I take an incomplete dominance, so in case of incomplete dominance, what will happen? This is also going to express 0%, this is also going to express 0%, and rather a new phenotype will emerge. Okay. So if I talk about the F2 phenotypic ratio, so in case of complete dominance, it is 3:1, but here it is 1:2:1. Here also it is going to be 1:2:1. As far as genotypic ratio is concerned, that remains the same, that is 1:2:1, and here also it is going to 1. So genotypic ratio is going to be 1:2:1. Here also it is going to be 1:2:1, and here also it is going to be 1:2:1. So that's all. Thank you.